Hi everyone, my name is Enrico Viperno, and in this video series, we'll be introducing fluorescence microscopy. I'm an applied physics graduate student, part of the Schnitzer group at Stanford University. In this first part of the series, we will be discussing fluorescence fundamentals, the physical principles behind the method, how fluorescence is used in biology, and implemented in neuroscience. And then we'll be looking at some of the details about how the fluorescent proteins, the indicators of neural activity, are transgenically expressed in mice, as well as virally. Then we'll also be looking at some of the dynamics of the two common types of neural indicators, calcium indicators and voltage indicators, as well as looking at the structure and mechanism of the calcium indicators and the voltage indicators. In this section, we will discuss about fluorescence fundamentals. A fluorescence molecule can typically be characterized by absorption and fluorescent curves, as drawn here. Absorption occurs when the fluorescence molecule absorbs a photon. This happens at a low wavelength or high energy light. Fluorescence occurs when the photon is emitted from this molecule, and this happens at a high wavelength or low energy light. The difference between the peak absorption and peak fluorescence wavelength is known as the soak shift. To look at fluorescence in more detail, we can draw a Jablonski diagram, which is what we will do here for a Mach 2-electron fluorescent molecule. A Jablonski diagram denotes electron energy levels and the transitions between these states. Here we draw the ground state, the first excited singlet state, the second excited singlet state, and the first excited triplet state. The ground state of this molecule occurs when both electrons are in the ground state and are paired or anti-aligned. In the first excited singlet state, one electron is in the ground state while the other is in the first excited singlet state, but paired. Same happens in S2. In T1, one electron remains in the ground state while the other is bumped up to the triplet state. However, these are anti-paired or aligned. The black lines, which are drawn here, represent the vibrational energy levels of each of these states. These are quantized and are at a higher energy than the first state. So to begin, let's draw some absorption lines. Absorption happens very fast and occurs from the lowest ground state up to the singlet states. And this can occur at different vibrational energy levels. Fluorescence occurs from the lowest first excited singlet state down to the ground state. Once again, it can occur at different vibrational energy levels of that molecule. To have a complete look of the Jablonski diagram, we also need to draw the non-radiative transfers. So the first is vibrational relaxation, which happens at high energy vibrational levels down to the lowest level. This is one of the reasons why fluorescence occurs at the lowest vibrational energy level of that state, because vibrational relaxation is much faster than the timescale of fluorescence, which is around nanoseconds. This can also occur at S2. The second non-radiative transfer is internal conversion, which happens from higher excited states down to the lowest excited state. Once again, this also happens faster than fluorescence and is why fluorescence dominates from the lowest vibrational energy level of the first excited state. Finally, there can also be internal conversion, intersystem crossing from the first excited state to the triplet states. And this leads to phosphorescence, which is when a photon is emitted from the triplet state down to the ground state. Phosphorescence is very slow, around the millisecond timescale. So that concludes this discussion of fluorescence fundamentals and the Jablonski diagram. In this section, we will discuss about fluorescence in biology. The first discovery of fluorescence in biology was in the 1960s when the green fluorescence protein was found in the jellyfish. This molecule fluoresces green light when exposed to blue light. The discovery and then development of this molecule eventually won the Nobel Prize in 2008. In this slide here, we show a side-by-side view of HeLa cells in light microscopy and fluorescence microscopy. While in light microscopy, the whole field is illuminated, in fluorescence microscopy, we can engineer different fluorescent proteins to fluoresce at different colors and to bind to different substructures within that cell, as shown here. Another advantage of fluorescence microscopy is that the non-fluorescent sample remains dark, and thus we can achieve very high contrast and sensitivity, allowing us to distinguish individual molecules within that sample. Fluorescence is used in neuroscience to image neural activity. Historically, the most common type of organism used in neuroscience is the mouse. The mouse is used because it shares 80 to 90% of their genes with humans, and the brain structure and function is similar to that of humans. Also, with the invention of genetic tools, we can manipulate the mice gene expression, thus expressing fluorescent proteins or also imitating human-like diseases within the mouse. Also, since mice are small and inexpensive, they are typically used in laboratory settings. And finally, they are considered ethically justifiable compared to non-human primates. The two main types of fluorescence imaging preparations in mice are a head-fixed and a freely moving case. In a head-fixed, surgery is performed under anesthesia, in which part of the skull is removed and replaced with a glass cranial window. This is paired with a mounting bracket, allowing for the mouse to be head-fixed to the optical table. On this optical table, we have the microscope, whose objective lens peers into the brain region of interest. The main advantages of a head-fixed preparation is that there is no constraint on the microscope weight and size, thus we have very high-resolution images of large field of views. However, the main drawbacks is that since the animal is head fixed, it restricts natural movement and induces stress on the animal. It also requires extensive habituation of the animal. The second case is a freely moving case, in which all the basic optical components of the microscope are resting on the animal's head. The exception is in the 2P case, in which the laser excitation light is brought in through an optical fiber. Also, the miniature microscope is connected via wires to a computer or DAC for data transmission and storage. The novel component of this drawing here is the GRIN microendoscope, which is a gradient refractive index lens which punctures the brain of the mouse and relays the image plane outside the skull. However, I should note that both of these surgical preparations, that is the glass cranial window and the microendoscope, can be swapped vice versa with both a head-fixed microscope and a freely moving miniature microscope. However, it is most commonly found for a head-fixed mice to have cranial windows and for a freely moving mice to have the microendoscope. As you might expect, a main advantage of the microendoscope is that we can peer deep within brain regions of interest. Also, another advantage of the freely moving mice is that we can image natural phenomena. However, because the microscope is limited on its weight and size, because it has to rest on the mouse's head, there is typically a low resolution images associated with freely moving mice. Finally, since the mouse is moving, there's also extensive motion artifacts and blurs which need to be taken care of post data collection. Fluorescent proteins can be implemented into mice into two distinct ways, transgenically and virally. For a transgenic mouse, it is typically done by breeding a transgenic line that expresses a driver and another transgenic line that is known as a reporter into a double transgenic mouse. The advantage of doing it this way is that we can increase the combinations of the double transgenic mouse since there's many types of driver and reporter mice. Starting a newly transgenic line takes around 1 year, and doing it this way saves a lot of time. The main tool used in transgenic modification is the Cre or flip recombinase, which is an enzyme which can cut and alter DNA at specific sequence sites. In the driver line, a promoter is used to promote the transcription of the Cre or flip recombinase downstream. This promoter can be cell type and location specific. On the other hand, for the reporter line, there's also a promoter which entices the transcription of DNA downstream, along with the Cre or flip activation sites, which are known as LOXP for Cre and FRT for flip. These surround a stop sequence, which stops the transcription of DNA downstream. After these activation sites, there is a gene of interest, in this case, the GFP. Now, when these two are bred, only in the cells of interest where the promoter is expressed, the DNA is actually altered. That is, everywhere in the mouse, the DNA is a combination of the driver and reporter line, but in the cells of interest, the Cre is transcripted and then translated into the enzyme, and this Cre then cuts the DNA as shown here and actually alters the DNA only in this region. The transcription and translation then ends in expressing GFP in those cells of interest. Two ways to genetically engineer mice are through viral vectors or viruses. The type most commonly used is the adeno-associated virus, or AAV, which, when implanted in the region of interest, implants its foreign DNA inside the cell nucleus. This DNA then takes over the cell's protein formation mechanism to express the gene of interest within the DNA, in this case, the GFP. Compared to transgenic line, this does not alter the cell's DNA. In fact, there are pros and cons to both methods. While on a transgenic line, the offspring has universal expression of genes, thus we can have a whole family of mice with the same gene expression. In a viral case, it is a much faster way of prototyping different genes of interest. A type of AAV commonly used is the vascular AAV, which has been recently developed, whose AAV is injected inside the bloodstream of the mouse, and this AAV travels to the brain of the mouse and crosses the blood-brain barrier to then express GFP everywhere inside the brain. Another type of AAV, which is extremely powerful, are the retrograde and antegrade AAVs. These are used to measure the neuron connectivity across the brain. For example, when these two viruses are injected in a region of interest, the retrograde virus travels up the axon and to the dendrites of the neuron, while the antegrade virus travels down the axon and to the boutons. You might expect of tying a GFP to the retrograde virus and an RFP to the antegrade virus, thus labeling green neurons which project away from the region of interest and red neurons which project down the region of interest. Finally, we can combine the two strategies, the transgenic lines and the viral strategy, in a hybrid case to increase the advantages of both. That is, we could have maintain the cell type specificity with the driver line by having a cell type promoter activate a Cre, but also maintaining the rapid prototyping of the AAV by tying the AAV to a GFP with the stop sequence that can be cut by the Cre recombinase or flip recombinase. The two main fluorescent indicators of neural activity are genetically encoded calcium indicators and genetically encoded voltage indicators. These indicators are constantly evolving to result in higher brightness and faster kinetics. And evolving, I mean engineering. As a result, the information of this slide represents a window into the current progress of these indicators. For instance, here is a picture of GCAM6F, 6 meaning the 6th version, F meaning the fast version. In any case, here we'll be discussing the differences in dynamics between these two sensors. To summarize, calcium indicators are a proxy for neural activity and measure only the action potentials within the cell. However, they have highest NR and brightness, which allows for a reliable measurement of neural activity, and are the ones most commonly used today. Voltage indicators, on the other hand, measure membrane potential differences and thus can measure subthreshold dynamics. They also have faster kinetics, however, they are not as bright and are noisier. In this figure here, we show these two different fluorescent indicators. Calcium indicators are located within the cell, while voltage indicators are located on the membrane of the cell. The result is a picture in voltage looks like a negative of those cells. Another thing to note is that due to the limited space of the membrane compared to the entire volume within the cell, there is a restricted number of voltage indicators one can place before altering the biological and physical properties of the cells. Speaking on the temporal dynamics of these sensors, we can draw mock dynamics for a stimulus. For instance, here on the left side of the screen, we have an electrode which could input a voltage spike and stimulate the neurons. In our region of interest, we will then optically image these two different indicators. So here is an example response. As you can tell here, the calcium dynamics are a lot slower, and the rise and decay time are of these values here, while voltage sensors respond much more rapidly. One thing to note is that an action potential is around 1 millisecond. Thus, voltage spikes typically can measure individual spikes. However, as seen here, for a multitude of spike trains, the calcium levels increases over time. The calcium signal increases over time, while the voltage indicator increases slower. This means that high SNR signals of calcium represent spike trains, not individual spikes. Finally, here we can look at some spatial dynamics of these sensors. So once again, the stimulus is on the left side of the screen. However, we are measuring signals at different regions within the field of view with these different colors. As you can tell, the calcium signal dies over time, while the voltage signal is still represented far away. This indicates that calcium signals are a response to action potentials within the cell, while the voltage signals can measure the subthreshold dynamics, thus allowing for spatial propagation of stimulus to be measured. Let's discuss the mechanism and function of genetically encoded calcium indicators. First, let's discuss the main cause of change in calcium ion concentration within the cell. This is through a synaptic transmission. In a presynaptic neuron, an action potential travels down the cell into the bouton, causing a voltage-gated ion channel to open to calcium, allowing for an increase in calcium within the cell. This increase of calcium ion concentration within the cell causes the exocytosis release of a neurotransmitter into the presynaptic cleft. In the case of an excitatory neuron, this neurotransmitter might be glutamate, which would then bind to a ligand sodium and potassium ion channel, depolarizing the cell, causing an action potential more likely. Well, for an inhibitory neuron, this neurotransmitter might be GABA, in which case it would bind to a ligand-gated chlorine ion channel, hyperpolarizing the cell, making an action potential less likely. So it is important to realize that calcium is a proxy or secondary messenger of neural activity. The GChem thus is an indirect measure of action potentials within the cell. There are two common types of calcium indicators, the most common being GCamp. GCamp is a chimeric protein, meaning a fusion of different proteins, these proteins being GFP, calmodulin, a calcium sensor, and myosin light chain peptide. GFP is composed of a chromophore, the molecule doing the actual fluorescence, and a beta barrel. In the presence of water, the chromophore's fluorescence is restricted. Thus, the beta barrel restricts the chromophore, surrounds it, restricting water molecules to access it, thus allowing for the chromophore to fluoresce. It is exactly this conformational change which the GCamp sensor takes advantage of. As calcium binds to the calcium indicator calmodulin, it brings in the myosin light chain peptide, thus closing the beta barrel around the chromophore, allowing it to fluoresce. This is a variable transition, allowing us to measure the relative changes in calcium concentration within the cell. Another style of calcium indicators are through FRET, or Forster resonance energy transfer. These are composed of two fluorescence indicators, a donor and acceptor. In the donor fluorescence indicator, it absorbs a photon and then through vibrational decay may decay to an energy state which can transfer its energy to the acceptor indicator, which could then fluoresce in a different wavelength. The efficiency of this energy transfer, is inversely proportional to the sixth power of the displacements between these two indicators, thus making this setup here extremely sensitive to small changes in displacements between these two indicators. When calcium is not present, the donor is far away from the acceptor, thus allowing for a high fluorescence, while in the presence of calcium, the myosin light chain peptide is brought closer together, thus allowing for a higher thread transfer efficiency, allowing for the acceptor fluorescence to increase. This change in fluorescence between the donor and acceptor can be extracted to relative changes in calcium ion concentration within the cell. There are many types of genetically encoded voltage indicators present. Here we'll be discussing three main types in my opinion: single FP voltage indicators, FRET voltage indicators, and opsin-based voltage indicators. All voltage indicators have in common the fact that they are placed on the cell membrane, thus allowing for the measurement of potential difference from outside the cell to inside the cell. In a single fluorescence protein, the voltage-sensitive domain opens and closes the beta barrel, restricting the chromophore, thus allowing for a variable intensity change of the green fluorescence protein. In a FRET voltage indicator, the voltage-sensitive domain brings together the donor and acceptance fluorescence indicators. And finally, a opsin-based voltage indicator is based on opsin, which are proteins that are light sensitive and form the basis of all visual systems in the animal kingdom. In our case, they are placed in the rods and cones of our retinas. They change their conformation from a resting state to a signaling state upon light absorption. And some microbial opsin have been found to be voltage dependent, thus allowing for the fluorescence of the opsin itself, or in the case of a dim fluorescence of the opsin, this can be attached to a FRET fluorescence protein. On a side note, opsins can be used in channel rhodopsin, which is a light-gated ion channel, thus allowing us to manipulate the action potentials within a cell. This field is known as optogenetics.